On this week's GARNet Research Roundup, I'm really delighted to be joined for a second time by Professor Malcolm Bennett from the University of Nottingham. And we're talking about a paper which has literally just been published in Science, and the title of which is Roots Branch Towards Water by Post-Translational Modification of the Transcription Factor ARF7. So this paper really brings together an old love and a new love of Malcolm. So the auxin response, which he's built his career on, and then the work about hydropatterning, which has really become much more important in the lab over the last few years. So thanks very much, Malcolm, for joining us again. And it'd be great if you give us an overview of the significant parts of this paper, please.
This is part of a long-term collaboration between. I really need to acknowledge the important input of several people. So ourselves, Ari Sadadonen's lab in Durham, and also Jose Dinany, who was really I suppose the father of hydropatterning. And it's a fascinating adaptive response. I love the way that roots are incredibly plastic to their environment, and hydropatterning represents a response where roots branch towards water, or as revealed in this paper, in fact, what plants actually do is they repress branching when they don't sense water is available. So it's a great system to actually study how plants sense water is available in the environment. Now, we knew nothing about how this took place. We knew from earlier work published with Jose that this was an auxin-dependent mechanism, but we didn't know anything about the underlying molecular mechanism. And this is really what the paper reveals. It reveals that when a root cells which are close to the tip are deciding whether to branch to form a new lateral root, a plant makes a decision very, very early on to actually whether water is available to it. And how it does it appears to be beautifully linked with the hydraulics. So the cells which give rise to a new lateral root are just at the positions just when the cells are leaving the root apical meristem into the elongation zone. And this is precisely where the root cells start to take up water. So it's beautifully positioned to receive information from the external environment to see whether water is flowing freely in or not. And we think that this is not just chance, this is deliberate. And this is where a plant will decide, okay, well, if there's water available, that means there are going to be nutrients available. And so why put your resources where there is no water? Because there'll be no nutrients attained. So we think that the mechanism is, that we revealed appears to be linked to the modification of a key protein. So one of these, I call them God genes, that these are transcription factors which sit at the top of the transcriptional cascade that give rise to lateral roots. And this gene called ARF7 appears to be modified in response to water stress. And the modification is something called SUMO. And SUMO is small ubiquitin-like modifier, so it's a ubiquitin-like protein, but unlike ubiquitin, what it does is it actually changes the properties of a protein. And so what we observed was that ARF7 proteins, which are made on the side of the plant exposed to water, are not modified by SUMO, and they just function as usual. And the ones which are exposed to the side of the root, which is dry, if you can use that terminology, or less water, they become sumolated. And what was fascinating, when you sumolate ARF7, this sumolated at several positions along the protein, one of which is in the DNA binding site, so it reduces DNA binding affinity, but it also results in recruitment of repressor proteins. And so effectively you're switching off the activity of the protein. And so you can create this schizophrenic regulatory behavior. On one side, it's highly repressed and not able to switch on gene expression. And on the other side, it's business as usual. And what was really cool, we managed to see using direct targets, reporters made to direct targets were able to see this asymmetric expression in the elongation zone. So we could almost see in real time, we could see this asymmetry happening if we put impose a water availability gradient in the elongation zone.
Well, this is one of the key aspects of the paper, the ability to image the lateral roots initiation in this dynamic environment. So can you say a little bit about the techniques you used which allowed you to image this, which might not have been able to be done previously?
Yeah, we were very fortunate to receive Alert 14 funding for a light sheet microscope.
Yeah.
And we could not have done these experiments without it. And also a brilliant postdoc called Daniel von Wangenheim, who really, he was trained by he was a PhD student of the inventor of the modern light sheet microscope. And this allowed us to do multi-view imaging and to build up a 360 view of lateral root development. And so we were able to see, using these direct targets of ARF7, using the light sheet, we were able to see exactly whether there was a bias on the one side or the other for where lateral roots initiate. And what was beautiful, at the very, very early stage of lateral root development, in the cells which give rise to the lateral roots, these are pericycle cells, we could see this asymmetric expression pattern. And it was biased towards the side which was exposed to water. And what was really nice, if we mutated ARF7 and looked at this reporter, and we could actually see that this asymmetric expression pattern disappeared, which was really nice. We also did the other experiment, which I think is far more important. We put the wild-type gene ARF7 back into the mutant, and we could see that it rescued the pattern. But if we mutated the simulation sites in ARF7, it couldn't.
Right.
So this provided definitive evidence that simulation of ARF7 was key to create this asymmetric pattern.
I see. Yeah, indeed. So it allows you to do this imaging in 3D as opposed to just growing things on a plate, which obviously gives you a false impression. The lateral roots kind of only can come out in two directions. Yes. So we know a lot about obviously this auxin, this kind of canonical auxin signaling between the relationship between ARFs and IAA proteins. Do you think, or do you have evidence that ARF7 is the only one which is involved in this? Since we know there's like 20 odd ARFs. So have you got any evidence that it's specific to ARF7, or is this a general response?
That's a really interesting question. We know that if we mutate ARF7, we have a hydropattern defect. If we mutate the other ARFs, we don't. So we can mutate, say, ARF19, its closest ortholog and homolog, and it doesn't have a hydropattern defect. Similarly, ARF6 and 8, these are all activating ARFs. So they're normally transcription factors which switch on gene expression. We can't do ARF5, the remainder, because it doesn't form roots. So it's a bit of a technical problem, that one. So we think ARF7 plays a really important role for hydropatterning. However, we do have evidence that these others are targets for simulation. So they can retain several other sites. And we have direct biochemical evidence, which we didn't publish in the paper. This is the work of Ari's lab in Durham. He's shown directly that ARF19 is a target for simulation. Now, we know simulation is actually controlled by a number of environmental stresses. Many proteins are targets for simulation. So it's a really hundreds of proteins are targets. Probably almost every protein is. Why does the cell simulate its proteins? Well, actually, what it offers is an opportunity to really change the interactome. So when you simulate a protein, you allow other proteins which contain so-called sumo interaction motifs to interact via so they're called SIMs, sumo interaction motifs, to interact with the simulated proteins. So you completely rejig the interactome. So when you typically find when you expose a plant to abiotic stress, and we showed this for ARF7, you see an enhanced simulation level. And that means that proteins which contain SIMs are now able to interact with other proteins which are simulated. And we were amazed to see that when this happens, ARF7 suddenly starts to recruit a very specific form of the AUXIAs, which is this protein called IA3 or SHI2. And this interaction between ARF7 and IA3 appear to be sumo dependent. So this completely goes against the dogma that ARFs and AUX-IAs interact through these domains 3 and 4. This interaction appeared to be sumo-sim dependent. And we showed that in a series of experiments, pull downs, and also in vivo expression, functional assays. So that's really interesting, because what you've got here now is an environmentally regulatory way to control the auxin response machinery. Because you can imagine you expose a plant to stress, you get accumulation of proteins, and this suddenly allows you to reconfigure auxin responses through recruitment of the IA3. So I've given this a name I call it ADEPT, auxin-dependent environmental perturbation of transcription.
Okay, there you go.
And I think that this probably is a mechanism which is used in other parts of the plant for ARFs, because in principle, if these other ARFs are targets of simulation, they should also recruit other AUX-IAs, but under different environmental circumstances. So I think this is a new kind of paradigm for auxin signaling, or regulation of auxin signaling through simulation.
Okay, that's great. So thank you very much, Malcolm, for giving us a brief tour around this paper. And there's no doubt that there'll be lots of extra work coming out of your lab on this topic in the future, I'm sure.
Thank you very much, GARNet. And I'd just like to say this was a brilliant team effort from Durham, and also US collaborators and collaborators in Europe as well. And it's an example of how our research is really laboratories without barriers, without walls. It's multinational. And also, we'd like to say a big thank you to BBSFC for supporting us 4 years ago. And it takes that long to deliver something like this. Not only the grant, but also the light sheet microscope. Without those investments, we could never have done this work. So we're very grateful.